Right. Today we are going to talk about antibody. This is the class number 2. And this class I will discuss about the classification of antibody, term related to antibody and function of antibody. Okay? What is mean by the classification of antibody? We have classified antibody into five classes: IgG, IgA, IgM, IgE, and IgD. So these are the five different classes of antibody that we will discuss in this class. Students, we have five types of antibody in our body. On what basis? We have classified antibody into five classes. This is only for the heavy chain. By the heavy chain, we have classified antibody into five classes. Okay. This is the heavy chain, and this is the also the heavy chain. This is the light chain, and this one is also the light chain. Antibody have two heavy chain and two light chain. There are, as you know, that heavy chain are of five types. Antibody have two heavy chain and two light chain. So, in heavy chain are five types. Heavy chain maybe gamma chain. Heavy chain, gamma heavy chain, alpha heavy chain, mu heavy chain, epsilon heavy chain, and delta heavy chain. These are the different types, five types of heavy chain. And light chain have two types: kappa and lambda. Kappa and lambda are the two light chain, while these are the five heavy chain. So those antibody which have gamma heavy chain, this is the antibody. Suppose this is the antibody. Suppose this is the IgG. Why it is called IgG? Because this antibody have gamma chain and as a heavy chain. Those antibody which have heavy chain as a gamma chain, it is that antibody is called IgG. While those antibody which have heavy chain as a alpha chain, it is called IgA, and those antibody which have heavy chain as mu, it is called IgM, and so on. So we classify antibody into five classes only by the basis of heavy chain. Our body have only five types of heavy chain: gamma, alpha, mu, epsilon, delta. These are the different five types of heavy chain. Each of the heavy chain is made of certain number of amino acid by a specific sequence. This is made by gamma is also made by the some number and is a specific sequence of amino acid. This one also, and so on. And our body have only two types of light chain. Kappa and lambda are the two types of these are the two types of light chain. So we define the antibody into five types only on the basis of heavy chain. Suppose this is the IgG, immunoglobulin G, which have heavy chain as gamma. This is another antibody, IgA. Why it is called IgA? It is called IgA because the heavy chain there is alpha, so it is called IgA. What is the rule of the light chain? Light chain are of two types. It may be kappa or lambda. Each antibody have two kappa chain or two light chains. Antibody never have one kappa chain light chain and other is a lambda chain. No, antibody have only one types of light chain, and the light chain and the antibody may be kappa or lambda, but both are not present. Okay, so we classify antibody into five classes only on the basis of heavy chain. Okay, this is the classification of antibody. Each antibody have each antibody each antibody have two heavy chain and two light chain. This heavy chain may be alpha, gamma, delta, mu, uh, but the light chain is kappa or lambda. So there are present 5 classes of antibody. Okay, this is the classification of antibody. Now another point, term related to antibody. What is the term that is related to the antibody? Monomeric antibody, dimeric antibody, pentameric antibody, tetrapeptide. So what is mean by tetrapeptide? As we know that this is the term number one which is used for antibody. Tetrapeptide. This is a this is antibody. As we know that antibody have uh, 4 chain, 2 heavy chain and 2 light chain. So it is a tetrapeptide. Antibody is a tetrapeptide because it have 2 heavy chain and 2 light chain. This is peptide number 1, 2, 3, 4. So what is monomer? What is monomer? What is monomeric antibody? Monomeric antibody is that antibody which have 2 heavy chain and 2 light chain. This one, this is the simply antibody which have 2 heavy chain and 2 light chain. Simple antibody is called monomeric antibody. Simply 2 heavy chain plus 2 light chain. Okay, what is dimeric antibody? Dimeric antibody are those antibody. Some antibody, as we know that some antibody work in single state, such as a monomer, while some antibody work in dimeric form, like this. Some antibody work in pentameric state, like IgM. This is the IgG. IgG. IgG work in monomeric form. This is the active form of IgG. Some antibody are present in dimeric form. These antibody are combined together by special types of protein which is called G chain. G chain. G chain is a specific types of protein which combine two antibody and make them functional. This antibody only work in dimeric form. Pentameric antibody are those antibody which consist of this is a antibody. This is a group of antibody. IgM. This is IgM number 1. This is IgM number 2. IgM 3. IgM number 1, 2, 3, 4, 5. These 5 IgM combine together and make a group which is a functional group. IgM cannot work in a isolated form. Suppose this is one IgM. This cannot work. This is inactive and is a separate form. But this will become active when they combine together by a G protein. This is G protein which is which combine together bio IgM antibody. So pentameric antibody, IgM antibody is the pentameric antibody. Uh, Only IgM antibody only work or activated during the pentameric form. Another term, valency. What is mean by valency? Valency mean that a single antibody or a group of antibody uh, occupy antigen. That how many antigen is occupied by a group of antibody or single antibody. Each antibody can only occupy 2 antigen. This is the antibody. Antigen is attached here between the light chain and the heavy chain. So one antibody, a single antibody, can attach to into 2 antigen. This is antigen number 1 and this is antigen number 2. So this is called valency. Single antibody is attached to only two antigen. So This is a monomer. This is a monomeric antibody. This is the antigen number 1, antigen number 2. So monomeric antibody is divalent, while the dimeric antibody is tetravalent. This is the dimeric antibody which is connected by the J chain. This is the J chain, and these are the antigen. Dimeric antibody are tetravalent. So this is the valency. Valency means that at how many antigen is occupied by a single antibody or a group of antibody. So this is the antigen. What is avidity? Avidity mean at how many points a single antibody or a group of antibody is attached. As we know that IgG is a monomeric antibody. This is IgG. This is IgG. So this is a monomeric antibody which is attached only to at 2 points. This is a bacteria. This is a bacteria. This is the cell membrane of bacteria. While these are the antigens. Well, these are the antigen which is present on the surface of the bacteria. So IgG is attached to the antigen number 1 and antigen number 2. So IgG is attached at only 2 points, while IgM, this is the antibody IgM number 1, 2, 3, 4, 5, and these are connected together by G chain. So IgM is attached to the bacteria at mini surface, at mini point number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So IgM is attached at 10 points. So this is a high avidity. IgM is high avidity antibody, while IgG is low avidity antibody, because this antibody is attached only at 2 points. This is attached to two antigen number 1, 2, and this is attached to 10 antigen. So this is the term. These are the term related to the antibody. Function of antibody. What is the function of antibody? Antibody work by two ways. One is complementary system and the other is direct action. Complementary system is indirect action of the antibody, while other is called the direct action. I am not explain complementary system because I have explained this complementary system as a separate class. What is the direct action of antibody? Antibody can destroy disease-causing microbe with the help of a system, with the help of a complete system, with the help of a complicated system is called complementary system, and this is a slow system. But direct action is the action of antibody which directly destroys the disease-causing microbe. Agglutination, neutralization, opsonization, precipitation, and so on. These are the direct actions of antibody. What is agglutination? Agglutination is a direct action of the antibody. This is a rapid, this is a sudden, this is a quick action of the antibody which destroy disease-causing microbe like bacteria. This is the IgM number 1, IgM number 2, 3, 4, 5. So IgM is pentameric. What is agglutination? Agglutination is a process, is a direct action of the antibody in which a clump is formed. There a clump of bacteria, a clump of virus, etc. is formed. There is a clump is formed. In agglutination process, a clump of bacteria, a clump of red blood cell, non-matched red blood cell, bacteria, etc. Is formed. It is in the form of clump. So this is IgM antibody. This is 2, 3, 4, 5. This IgM antibody is combined together by J protein. Agglutination process is mainly occur by the IgM. This process is not controlled by the IgG because IgG is a monomeric, and in agglutination process a clump is formed. So for the formation of clump, IgM is needed. This is the antibody number 1. This is a bacteria. This is bacteria number 1, bacteria 2, 3, 4, 5. These are the bacteria. Red color, this one, this, and these are the antigen present on the bacteria. This is a bacteria. So this IgM group of antibody is attached to this point. This is attached to this antigen. This is attached to this antigen. This and this. So there is also attached another IgM. There present. It's a J chain. Okay. So agglutination is a process in which a clump is formed. By the bacteria, by the non-matched RBC, etc. So this is the agglutination process, which is mainly controlled by the IgM. Okay. Another process, opsonization. What is mean by the opsonization? Opsonization is a process. This is the direct action of the antibody. Especially this is the function of the IgG. This is the IgG. This is the IgG. Opsonization is the process by which a bacteria or other disease-causing microbe is engulfed by the macrophage with the help of the antibody which is present on the surface of the macrophage. This is a macrophage. This is a receptor on the surface of macrophage. This one is the receptor, while this one is called the IgG antibody. This macrophage have specific types of antibody which is called IgG antibody. Then this antibody is attached to the bacteria. This is a bacterial cell. This is a bacterial cell which have a specific site. This is a antigen and the bacteria. This is a bacteria. When this antibody is attached to this bacteria, then it attract this bacteria toward the macrophage, and therefore bacteria is engulfed by the macrophage. This is the destructive part of the bacteria. So bacteria is a die out. What is opsonization? Opsonization is a process, is a direct action of the antibody in which macrophage engulfed bacteria or other disease-causing microbe with the help of antibody. This is the function of IgG antibody. What is neutralization? What is mean by neutralization? neutralization is a process. This is a direct action of antibody in which a toxin or bacteria or a virus is neutralized, mean stop its function. Suppose this is a toxin or a virus, which in suppose this is a virus. This is a virus. This is the membrane of the virus. This one red color is the antigen present on the surface of the virus. Okay, this is the antigen. This is a human cell. This is a human body cell. Human body cell have a specific receptor for the antigen of a virus. This is a human body cell have a specific receptor. This is a receptor. This is a specific receptor. This receptor is combined together with the antigen present on the surface of the virus. This is the receptor for toxin, or this is the receptor for other antigen. So this substance, this antigen is attached here and make our cell is destroying. But antibody come and cover this antigen. This is antigen. So antibody come and cover this antigen, also cover this one. So the antibody cover this antigen. So the this virus or this bacteria or this toxin is cannot attach here with the help of this antigen, because this antigen is covered here. This is an antigen which is covered by the which is covered by this antibody, and then no long attachment is occur. So these are the function of antibody.